Welcome to wastewater treatment plants and anaerobic digestion. So just a primary overview of the wastewater treatment process. So it starts with primary treatment. So the incoming sewage gets in, you get out the grit, the sedimentation, the what settles goes. It could go into a digester, it could just go straight into sludge drying beds, and that biosolids then gets deposited in a field. Usually, sometimes they can even pelletize and landfill it. There's a lot of things you can do with that sludge, but it's basically all the solids get separated out. The liquid portion continues to aeration, another sedimentation to get the remaining solids, disinfection, and then it's put into the rivers, the lakes. Again, wastewater treatment plants are usually on a river. In DC, we're on the Potomac, and so they're put there. Now, if you're not on a river and you're not where water, then you have other types of land application that we'll do for liquid waste, and we'll review those later in the semester. So that was the easy one. Here is the actual very detailed process at the DC Blue Plains wastewater treatment plant. So you can see again that the waste comes in, you have the screens, you have prompting first you have an aerated grit chamber that gets rid of the grit, this primary settling tanks, gravity thickening sludge, and then you have a blend tank where you then take the secondary reactors where you aerate it. Again, you have dissolved air flotation and thickening to again get those sludges out. Then again, more aeration, nitrification and denitrification reactors. And then you go to those tanks. Again, you add disinfection. You have a filter at the end. And then finally, you discharge to the Potomac, while the solids that came through here go into this CANBI process, which we'll talk about, into anaerobic digestion. They're dewatered, and then they have land application. And the electricity then from this goes into cogeneration, a combined heat and power generator, a CHP, where you get electricity to run the plant, and the heat goes back to the CANBI and anaerobic digestion process. So that's the overview of the DC water system. So going step by step, you start with the bar screen. The bar screen is just going to catch the large objects that got into the sewer, bottles, bricks, pieces of wood, just the big stuff. Then it goes into the grit chamber. So the grit chamber is going to remove the smaller stuff, but stuff that we don't want going into our anaerobic digestion system and being field applied through the biosolids. So these are going to be things like rocks and gravel and broken glass, as well as diapers, cones, towels, plastic bags, syringes. You'd be amazed what gets thrown into the sewer and down the toilets. So again, that bar screen and that grit is then going to go to the landfill, okay? And only this velocity slows, and then it's going to go into our actually settling of solids for the biosolids. So our primary treatment, this is where we get most of our solids to settle. And this is where it's basically slowed down. The suspended solids settle down to the bottom by gravity. And that's referred to as our sludge or our biosolids. And then the floatable solids are also removed. And so we have a rotating kind of plow at the bottom here that kind of gathers all those solids as they go and puts them down the center, which again, they go to sludge treatment, which again can be composting, can be anaerobic digestion, can just be lime stabilization, various things we can do with those solids. And then the fats and oils are skimmed off the top. So in this, and at least a DC water, they also add ferric chloride and a polymer, and that actually helps with those solids settling. It gathers some of those solids and helps them settle down. So those settable solids is about 50% of the solids are going to settle out in the primary treatment. And about 35% of our carbonaceous biochemical oxygen demand, our carbonation BOD, is going to be removed through this primary treatment and the settling of solids. So the removal is really just settling out the solids. We're not actually doing a lot of biological treatment here. We're just separating solids from liquids, and we're using polymers to help with that separation process and settling process. And then the overflow then goes to our secondary process, with the solids going to a thickener, our thermohydrolysis processing and digestion. So this is what primary treatment looks at DC water. So here we have our primary treatment tanks. Again, they look very much like this. You can see there's the water enters here, and we have these plows there at the bottom, which you can't see, but again, it's very slowly rotating. and settling out the solids into the bottom of the system. Then we go to secondary treatment. So this is where we actually grow aerobic bacteria and other microorganisms to consume the carbon. So this is where we actually have metabolism going on. So they're going to break out those sugars, starches, carbohydrates, all those short chain carbohydrate compounds. The microorganisms are going to use those, and they're going to respire CO2, and they're going to degrade that organic matter. So this is where we get a lot of our BOD reduction. And then these microbes are actually recycled in this process. And then again, we monitor the total suspended solids, our BOD and our DO. And again, any additional sludge can be removed, while some of it is actually rotated to get our microbes back in. And this is a picture of secondary treatment. See the aeration, all the pumps. And so this is what's being aerated. And again, our microbes are doing it. And then we're going to recycle those microbes through the process as we treat our sludge and break down the carbon through microbial degradation with aerobic microorganisms. So again, we have, our flow. We're going to add ferric chloride here. What we're going to do here is we're trying to reduce, again, this ferric chloride has a two thing. Not only does it help with settling, but it also combines with the sulfur to reduce the amount of hydrogen sulfide that might be released, which can cause odors. So it's actually twofold. It actually reacts with the phosphorus and precipitates those phosphorus, but it also reacts with the sulfur to reduce the sulfur emissions. And then those aerobic bacteria can consume these solids that kind of precipitate out. Again, we're going to have more polymers that actually are going to settle out our bacteria to kind of keep them going with the cleaner water than going over our weirs. Then in DC water, they have tertiary treatment. Again, some wastewater treatment plants, depending on where they're located in the receiving bodies and their size that they're located within, they may have only primary treatment. They may have primary and secondary, or they have my primary, secondary, and tertiary. Our tertiary treatment is where we really concentrate on that nitrogen. So this is where we have our nitrification and denitrification processes. And so here we're going to add some sodium hydroxide. We have our air mixers again, because this is an aerobic process, our nitrification. And so the aerobic bacteria consume the ammonia and convert it to nitrite and nitrate. And then it's going to flow to an anaerobic anoxic tank where we're going to do that denitrification. Now, by this point, we've actually reduced a lot of the carbon. A lot of our BOD is gone. This is tertiary. So this is just for nitrogen. So we're actually adding methanol. And what that is methanol is just a small chain carbon. So we're actually adding, we usually get methanol from greenhouse gas production. We use greenhouse gases to produce ethanol, these small chain carbons. And then we use that methanol to give a very easily degradable carbon source for our denitrifiers to make sure they can denitrify because again, a lot of the carbon's been used up, and denitrifiers actually need a carbon source, and then they use the nitrate as their electron source. So we add methanol, and then we get nitrogen gas being produced. And then we're going to flow to another sedimentation tank to again get out that bacteria and recirculate it. And then we're actually going to have a filtration, and then we're going to put it into the river. So again, these are final clarifiers that you may have after this process. And again, and you might have this instead of tertiary treatment as well. And then our sludges, those are our biosolids. So the sludge, if you're just basic sludge disposal or sludge utilization, you're going to just do lime stabilization. All you're doing is you're adding lime, which actually increases the pH to kill off pathogens, and then you can land apply it. Or some people actually just landfill the sludge. Others incinerate it. You can digest it, or you can compost it. So the biosolids that come out, they're subject to pathogen limits. So again, these biosolids, by having these, so all the rest of the organics and nutrients that come into a wastewater treatment plant get, again, CO2 goes up, N2 gas goes up through nitrification, denitrification. This is actually the part of the wastewater process where we can put it back onto the fields to grow more food and get those nutrients back to where they can grow food. And we eat the food, we defecate, and then we bring the cycle back. If it's class A biosolids, That means that it's been at least 160 degrees for 20 to 30 minutes, and and the pathogens or indicators organisms are reduced to below detection limit with fecal coliforms less than 1,000 MPN per gram, most probable number per gram. If it's class B, then it's pH for at least 12 to a 12 pH for 2 hours. We keep it up there for at least 2 hours at 12 pH. And so we added calcium hydroxide for that. And so then the fecal coliform just has to be less than 2 million coliform units per gram. So this is what Blue Plains used to do before they added the digester. It was class B biosolids. Now with the digester, they are class A biosolids. And so what they do before the digestion process is, and this isn't true of all wastewater treatment plants, can be as a trademarked process where they actually do thermal hydrolysis. You can almost think of it like an Instapot, right? They have these solids that come in, they preheat them to almost 200 degrees Fahrenheit. And then, and they use steam. And then, and that's their pulper. And then they have a batch process where then they heat it up and they put it under pressure for 30 minutes. And then they have this flash tank where then after it's been under pressure, they depressurize and it destroys the cells walls and it cools down. And then it goes into the digester more at the 120, 100 degrees Fahrenheit that the digesters like we have methanogens in our stomach, digesters like around 100 degrees Fahrenheit, which is the temperature of our bodies, and about 8 to 12% total solids to go into the digester system. So here are the digesters. Again, here's the Canby process at DC Water, and here are our digesters. You can actually see them from the highway. And again, the energy is produced. They have about, again, 9 million BTUs per metric ton of biosolids that's produced. And then about 30% of this is actually available and utilized to make energy. They have about a 30% conversion rate. And so DC processes about 370 million gallons of wastewater. And then it is the largest thermal hydrolysis plant in the world. And so they produce approximately 13 megawatts of electricity, which is enough to power 8,000 homes. And again, they could power in even more if they brought in more offside food waste, but they don't do that at this time. But actually, a lot of their savings is not even the energy production, it's the trucking costs, because they've reduced the amount of solids through that anaerobic digestion process. So they have less that they're trucking onto the fields. So what is anaerobic digestion? So these are actually anaerobic digesters off of in Boston, Massachusetts. And here's like a low cost anaerobic digestion in Costa Rica. So they can be at various scales, very little, very big, but anything can go into them. It can be manure, crops, wastewater, sludge, food waste, anything that has organics. And then it's a stepwise process to break down those organics into smaller chains, which are finally used by the methanogenesis. So it's a 4-stage process where they produce methane. So methane is the biggest part of the biogas, but there's other constituents as well in this gas that's produced from this process. And then what comes out is again, you can think of it as taking broccoli or manure or something that's very solid, and it comes out like a brown tea. So you reduce those solids. The carbon that was there has been going to smaller and smaller chains and then goes up to CH4 and CO2. But your nitrogen, your phosphorus is now liquid. And so that nitrogen phosphorus at a wastewater treatment plant can actually be put back to the front of the wastewater treatment plant to go through the wastewater treatment plant processes. And then there is some nitrogen phosphorus in the solids as well that are left over. And so you can still use those nutrients to apply into the field if it's a farm-based digester, or you can bring them back to the front of the wastewater treatment plant if it's a wastewater treatment plant digester. So again, our lignin is not gonna do much for us, but our celluloses, our starches, and our proteins, they're gonna be broken down to our monomers, our amino acids, which are then gonna form during acetogenesis, form our volatile organic acids, acetic acid, propionic acid. And then through methanogenesis, those are gonna form our methane, CO2. And so again, there's some nutrients used for microbial growth, but most nutrients go through. And again, our rate limiting is really kind of the breaking down this first hydrolysis fermentation process, the breaking into those small chains that can be used by the microorganisms. So again, this is just another look at the same thing. We have complex polymers. We have that hydrolysis process, which is often rate limiting. And then we have our monomers, our sugars and fatty acids, which get to our acetate, which again, we go through acetogenesis, which is acetotropic methanogens, which then produce that methane. And that's about 70% of the methane in digesters. But we also have another group called hydrogenic methanogens. And these actually use, it's about 20 to 30% of our methane produced, where you combine H2 and CO2 together to produce CH4. So again, there's two processes going on, creating those acetate, those small chains used by acetotrophic methanogens, or hydrogenotrophic methanogens using H2 and the CO2 that's produced already in the digester and combining that together. So the methane can be anywhere, if you think of biogas, anywhere from 55 to 68%. The rest is pretty much CO2. Less than 1% usually is hydrogen sulfide, ammonia, and then water vapor. End uses, I can use it in my kitchen for cooking. I can spray the fertilizer onto the fields, again, in small scale here in Costa Rica, or through our machinery in the US. I can put the biogas through a digester. I can carry the biogas to where I need to use it. This is actually in Vietnam, and use it for cooking. So often we'll do a digester where we do a cogeneration unit, a CHP, a combined heat and power, so that we can heat the digester with the extra heat from our electricity production. And then again, we can do the electricity. Often if it's even in a wastewater treatment process, we're gonna do solid separation. If it's on a farm, then those solids are gonna go to the field. On the farm, those liquids would also go to a field. At a wastewater treatment plant, those liquids are gonna go back to the front of the wastewater treatment process. So if we're thinking about designing a digester, we need to think about the characteristics of our waste. What is the organic matter? How much ammonia, volatile solids, sulfur? What's the solid and liquid retention time? Are we solid liquid separating after digestion, before digestion? What's the hydraulic retention time? Again, in a normal wastewater digester, it's going to be about 20 to 30 days. Same thing for a farm-based digester. If you have more complex substrate, like food waste that needs to break down, or even crop waste, something that's harder to break down than manure or sludge, which has already partially been broken down, then we might need more than 20 to 30 days. Again, the temperature, it can be at two ranges: mesophilic, which is the range in our body, about 100 degrees Fahrenheit, about 35 degrees Celsius, or thermophilic, which is about 55 degrees Celsius. Again, we need to think about the pH and the alkalinity, and the methanogens like a neutral pH. The presence of any inhibitory substance that might actually, if we have a lot of chlorine, especially a small digester, if you're putting a lot of bleach down the toilet and it's a very small digester, that may overwhelm the good bacteria that we have in there for making methanogens. We need to think about, again, ecological design, thinking about the macro and micronutrients. We don't want too much ammonia, or that can be toxic, but we want to make sure we have enough, of the micronutrients that they need, the boron, the molybdate. We have to think about hydrogen sulfide production and treatment, because again, that hydrogen sulfide, even though it's less than 1% of the biogas, it can be toxic to the utilization of our generators. How much biogas are we going to get so we can size our generator, and how are we going to heat, and then how are we going to use our biogas? Again, methane is a very potent greenhouse gas, and this reduces the amount of methane emissions. We actually capture that methane and use it as energy. It controls odor. We stabilize manure, and we decrease wastewater treatment costs. And then if it's co-digestion, we can actually reduce the amount of food waste going to landfills. If we think about them specifically in the wastewater treatment context, again, about 150 of the 16,000 wastewater treatment plants in the US used it. Less than 10% of where combined heat and power is technically feasible. Currently, wastewater treatment plants count for 3% of all the electricity use. Literally 3% of our electricity in the entire US goes to treat our waste. And the rule of thumb is it takes about 0.5 kilowatt hours to treat 1 meter cubed of 1 cubic meter of wastewater. Wastewater plants in the US also generate about 7 million tons of biosolids per year. Most of this is generated in large cities, and they must be managed in a way that protects waterways and also increase food production. So if we think about wastewater treatment plant energy consumption, a lot of it's with this pretreatment. We have pumping, we have deodorization, mixing. This is where we use a lot of the energy. Our biogas cleanup, again, once we produce the biogas, we want to remove moisture, remove H2S, and we could remove CO2 to create renewable natural gas. To remove moisture, we're going to use heat exchangers to cool the biogas, and then that condensate will be removed. To remove hydrogen sulfide, again, if we're just using it in a boiler for heating, we can have about 1,000 ppm. If we're using it for an engine generator or combined heat and power, we want to be less than 500 ppm. we're using it for pipeline injection, renewable natural gas, we need to be less than 4. So how can we remove our hydrogen sulfide? We can do it using the chlorine dosing, which we do at wastewater treatment plants. We can do what they call iron sponge, or we can do a microbial fixation process. So if we look at microbial fixation, again, we have hydrogen sulfide. We actually add a little bit of oxygen, and then we use a bacteria called Theobacillus, which actually is able to oxidize it to sulfate. So if we think about biological remover, we can actually inject air into the headspace of the biogas. And then that oxygen that's in that air actually helps to reduce the H2S there in vessel. And then the sulfate, it goes into the water and goes out of the digester. So if we think about the biogas headspace, we can get actually this biobacillus actually on the headspace here, and then it actually produces it there. Or we can create a separate vessel where we have that biobacillus and we inject a little bit of air into our biogas. We don't inject too much because, again, air has nitrogen too. So we want the amount of oxygen to match the amount of hydrogen sulfide that we're trying to reduce. To remove biogas, we can actually do various steps: regenerative water wash, regenerative mine wash. Pressure swing absorption is probably the most common. And again, we're trying to remove that CO2, so it's now 100% methane, which is natural gas. We can also do membrane separation or cryogenic distillation. All right, thank you very much.